Hello, everyone. I hope you're doing well. This is Mr. Caudell. I'm doing well. Um, Here we go with meiosis. So we've learned about mitosis, how cells divide. Now we need to learn about how sex cells, the sperm or the eggs created in the testicles or the ovaries, are created. It is different than mitosis for sure, and it allows for the variation which exists in any population of higher organisms, like cats or like humans. There's a ton of variation, and that's good because if the environment changes, which it always does. There's ice ages, there's heat waves, there's new viruses that come into environments. Lots of different things occur, and some organisms are better fit to survive in that environment than others. And the reason why is through variation in a population, and that's because of meiosis. Meiosis creates variation amongst egg and sperm, and so any person that's ever created on this planet between a union of two people, even within families, is not even close to being nearly the same as someone else is in that family or on our planet. In fact, they say that for every guy creates like something like 12 billion sperm in their lifetime or something, some crazy number, and they'll never create the same exact sperm amongst all of those. There's so much variation within the sperm. And so the same goes for eggs. So here we go with meiosis. I want to show you the bigger picture first. Remember, we have these haploid sex cells called sperm and eggs. They each have 23 chromosomes in them. 23 from dad, who's created the sperm, 23 from mom, who's created the egg. When they come together, they create a zygote. Let's imagine that zygote divides millions of times. Finally, it's puberty, and someone's going to start to, girls create their eggs even before they're born. But let's imagine a guy, for instance. Finally, down in a guy's testicle, there just has been cells that are just diploid in nature, like every other cell in the body down there. It's been growing, and then all of a sudden it gets a signal, hey, let's create some sperm. So this is what's different. So there's 46 chromosomes in that very first cell that's going to create a sperm or an egg, just like any other cell in the body. But then it undergoes, instead of mitosis, it undergoes meiosis. And there's two phases to meiosis, meiosis 1 and meiosis 2. You can see that here. At the end of meiosis 1, at the end of that first division, you have cells that still have 46 chromosomes in them. But there's a big difference here between this and mitosis. These are not the same, and these are not the same as the parent cell. These are called daughter cells, and that's called the parent cell. And I'm about to show you what happens in meiosis 1 to make these not the same. And then these will then further divide down to create this haploid number that we saw up here. And that's important because you need to only have half the number so it can combine with, like an egg, for instance, to make a diploid cell that we call a zygote. None of these are the same. I'll just write it down here, not the same. And that's on purpose, as I just tried to mention. I want you to look for two things: crossing over and independent assortment. These things are the things that occur in meiosis 1 that give cells these developing sperm or eggs their differences between one another. Well, I guess while we're here, I might as well show you that in the textbook on page 323, there's some information on meiosis, and I'll probably have you read this. Sorry about that. There's lots of good pictures in here. It's kind of dense with information, so make sure you spend some time on pages 324 and 325. And then you're invariably going to have to compare mitosis and meiosis like this table does. Just looking for the number, figure 11-17, and then over here there's some more written information about it. Well, this is the state standard at the very top of this turn-in sheet. Now, when you read this for the first time, it doesn't make any sense. I'm going to read it to you. It says, evaluate data showing that offspring are not clones of their parents or siblings due to the meiotic processes of independent assortment of chromosomes crossing over and mutation. So there's a lot here Remember, I told you to look for these things. The reason why I said what's up on this is because it's hard to kind of unpack this, but we're going to do it real slowly. It's true, if you read the green here, that any offspring, let's say a son or daughter to a set of parents, are not clones of their parents. That's true. And they're not clones with their siblings. And the reason why is because of meiosis. And these meiotic processes of independent assortment and crossing over, and then it says mutations on here. And mutations are always happening in DNA. This is state standard 2.7D. So just be patient with it and we'll be able to get through it. You should have a handout here. It's got 4 pictures on it. I want you to start with the comparison of mitosis and meiosis at the bottom on my handout. Go ahead and put a vertical line down the middle of this. These are 2 separate processes. I would ask you to press pause and go through each of these and try to remind yourself what's happening in mitosis and take a look at how mitosis is similar or different than meiosis. Well, in the creation of these sex cells, and you can see here a sperm trying to penetrate an egg, many sperm. You have to understand that right here, before these kind of double chromosomes will split, the whole double chromosome gets pulled to one side. I'll show you that in a second. But the point is that in meiosis 1, what happens is you have these chromosomes from mom and from dad. Remember, they came from up here and now they're here. And you've got a number 6 from mom and a number 6 from dad, a number 12 from mom, number 12 from dad. You have a number 3 from mom and 3 from dad. I just put some arbitrary numbers on here. And the idea is that these are called homologous chromosomes because they're similar. Maybe right here's the gene for eye color. I've been using that example. And you see here that they've doubled from a single chromosome to 2. And now these are called sister chromatids. So there's 4 chromatids here. There's a sister chromatid pairing of this doubled super chromosome from dad and one from mom. And you can see that they're kind of similar. And because they code for the same characteristics, but not exactly like height, we've used that example, or hair color or whatever. It says hair color right here on these 2 chromosomes, one from mom, one from dad. But it might say something a little bit different. Hey, blonde hair or dark hair. But because the characteristic is the same, you can see here that they're kind of like overlapping. They're going to actually break off pieces of themselves and swap those. And then you can see that down here. We'll get to this detail in a second, but I just want you to look for this. You see how there's now a different color on these chromosomes? What has happened here in this instance is some have broken off. And so some of the genes that were once on mom's chromosome are now on dad's chromosome and vice versa. That's called crossing over. We'll get to that detail. And then we'll get to this independent assortment in a second.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Okay, so you have a handout, as I've shown you before, and you got this picture up on the top. And what you're looking at here are these homologous chromosomes that are kind of getting up close to one another, and they're doing something called crossing over. So here's our homologous pair. It's up there on the screen or on your screen, and I have it up here on the board. This is called crossing over. This occurs in prophase 1 of meiosis 1. Remember, I told you that meiosis has two parts to it, meiosis 1, meiosis 2. Prophase 1 of meiosis 1 is where this occurs. You can see how they're kind of like squiggling over each other on the board and again on the screen. You can see now how they've swapped over genetic material. It's just what happens. It happens in testicles and ovaries. I'd like to say it happens in guys' testicles all day long, but in girls' ovaries, interestingly, you already did this before you were even born. You did all this meiosis 1 stuff, and then the whole process of creation of eggs like arrests or stops before you're born. You can see here prophase 1. I'll go ahead and flip it over and put a horizontal line right here going across. And you can see prophase 1 of meiosis 1. This is where crossing over occurs. You can just write that down on there if you like. There's crossing over. Well, you can imagine then you're going to create chromosomes that have never been created before. I'm pointing to the board, but if you look up there in the bottom left corner, you can see kind of these funky chromosomes. They randomly get broken off. You have to remember these things are hundreds of millions of letters long, and so they get broken off at even just one nucleotide letter different than the last time. You're going to have a new chromosome that's never been created before, and then that's going to go on through this process, and you're going to create some sperm or some eggs that have never that are unlike any other sperm or egg that have been created, which will then create babies that are unlike any other that have been created. So it's pretty cool. Remember, the goal here is to make variation through meiosis. We've evolved to have this process. Here's the other process by which this variation is created in the creation of sperm and eggs. And if you look here, you can see that when the cells are getting ready for the first meiotic division, and you get this metaphase, just like in mitosis where everything lined up on this plate, this metaphase plate in the middle. These guys it's a little bit different. They're going to line up on the sides of the metaphase plate. Now, if the blue chromosomes equal dad, chromosomes from dad in this process, and the red chromosomes are, let's say, from mom, who's to say which one's going to line up on the right or the left as we're looking at these developing sex cells of the metaphase plate? And the answer is that it's random. In fact, since there's 23 pairs here, and there's two different ways that they can line up, 2 to the 23rd is 8.4 million variations of how these things can randomly line up at the metaphase plate. You can see on the left side, you've got all of dads on one side and all of moms on one side, and then the right one next to it, you've got one of dads and 2 of moms on the left, and so on and so forth. You can just see how they can line up randomly like that. Well, you can imagine that when these things then split in order to create the 2 sperm over here, and then this guy is going to create the 2 sperm over here, depending on how they randomly lined up during this process, you're going to get totally different combinations of sperm of genes from either mom or dad. So, some people say, oh, you look more like your mom, or you look more like your dad, or you have this characteristic of your mom or your dad. Well, it's because maybe the egg or the sperm that was created had more of that. I guess it would be grandma and grandpa, if that makes sense, because if a sperm is being made, like one of these sperm that's going to create you, it's really grandma's and grandpa's genes or chromosomes that are going to be pulled into these respective sperm in a random manner. This is called independent assortment at metaphase 1 of meiosis 1. Happens right here. Go ahead and write metaphase 1 of meiosis 1. I think you should press pause, and I think that you should right now talk to your favorite stuffed animal or your neighbor about crossing over and independent assortment and how those things lead to variation. Additionally, when you're ready, get a blank piece of paper. I would give you one if you were in class, and I would just say copy all of this down. I want you to have this in your notes. So let's do that. Go ahead and press pause. Just take a few minutes and copy all of this down. Write your own notes on it, just so you understand. Well, I guess we'll go back. You can see here, you've created a horizontal line here. This is meiosis 1 that's happening at the bottom of the page, equivalent to this. And then meiosis 2, the second half of it, is happening up here at the top of the page. So meiosis 1 has some really quirky things. And then once meiosis 1 is done, meiosis 2 occurs. And it's just the second half of this process. You can write down next to it, like mitosis. Meiosis 2 is very similar to mitosis. And the way things go, let me show you that. And the way things are like yanked around, it really looks very similar to mitosis, but you have kind of these funky looking chromosomes with little broken off pieces. But other than that, it looks exactly like mitosis. I'm going to show you a 6-minute movie, and then we'll be done.
Reproduction, always a popular topic. One that I don't mind saying that I am personally interested in. The kind of reproduction that we're most familiar with is, of course, sexual reproduction, where sperm meets egg, share genetic information, and then that fertilized egg splits in half, and then those halves split in half, and so on and so on and so on to make a living thing with trillions of cells that all do specialized things. And if you're not suitably impressed by the fact that we all come from one single cell and then we become this, then I don't know how to impress you. But riddle me this, my friends. If sexual reproduction begins with sex cells, the sperm and the egg, where did the sperm and egg come from? Oh, dude. So how do sex cells form so that they each have only half of the genetic information that the resulting offspring will end up with? And for that matter, why aren't all of our sex cells the same? Like, why are my brother John and I different? Sure, we both wear glasses and we both kind of look like a tall Doctor Who, but, you know, we have different color hair, different noses, and I'm way better at Assassin's Creed than he is, so why aren't we identical? As far as we know, we both came from the same two people with the same 2 sets of DNA. The answer to these questions and a lot of other of life's mysteries is meiosis. In the last episode, we talked about how most of your cells, your body or somatic cells, grow themselves through the process of mitosis. Mitosis replicates a cell with a complete set of 46 chromosomes into 2 daughter cells that are each identical to each other. But of course, even though the vast majority of your cells can clone themselves, you cannot clone yourself. And for good reason, actually reasons. If mitosis were the only kind of cell division we were capable of, that would mean A, you would be a clone of one of your parents, which would be awkward to say the least, or possibly B, half of your cells would be cloned from your mom and half would be clones from your dad, and you would look really weird. But that's not how we roll. We do things a better way, where all of your body cells contain the same mix of DNA, 46 chromosomes grouped. That's 23 pairs, 1 in each pair from your mom and 1 from your dad. Those pairs of chromosomes are pretty similar. They're not identical. They contain versions of the same genes or alleles in the same spot for any given trait. Since they're so similar, we call the pairs homologous chromosome pairs. Homologous is a word that comes up a lot in genetics. It just means that 2 things have the same homo relation logos, even if they are a little bit different. However, there are some very special cells that you have that have only 1/2 of that amount, 23 chromosomes. Those are sperm and egg cells. These are the haploid cells. They have half of a full set of chromosomes. And they need each other to combine to make the complete 46. Creating those kinds of cells requires a process that's very similar to mitosis, but with a totally different outcome. Meiosis. That's when specialized diploid cell splits in half twice, producing 4 separate cells, each of which is genetically distinct from the others. Meiosis is a lot like mitosis, except twice. It goes through the same stages as mitosis: prophase, metaphase, anaphase, and telophase. Then it goes through another round of the stages again, and they have the same names, conveniently, except with a 2 after them. They're like sequels. And just as with the Final Destination movies, the sequels have pretty much the same plot, just some new actors. So the raw materials for this process are in your ovaries or your testes, depending on, you know what it depends on. They're diploid cells called either primary oocytes or primary spermatocytes, depending on what kind of gamete they make. Men produce sperm, you may have heard, and they produce it throughout their adult lives, whereas women are born with a certain amount of eggs that they'll release over many years after puberty. Here you might want to go watch the previous episode about mitosis again, because that's where we go into detail about each stage of the process. Once you're done with that, we can start making some baby makers. So just like with mitosis, there's a spell between rounds of cell division where the cell is gearing up for the next big split, and this is called interphase, when all the key players are replicating themselves. Long strings of DNA in the nucleus begin to duplicate, leaving 2 copies of every strand. To jog your memory about how DNA does this, we did a whole episode on it. You can watch it and come back. A similar process takes place with centrosomes, a set of protein cylinders next to the nucleus that will regulate how all of the materials will be moved around along these ropey proteins called microtubules. And that brings us to the first round of meiosis, prophase 1. This is nearly the same as in mitosis. The centrosomes start heading to their corners of the cell, unspooling the microtubules, and the DNA clumps up with some proteins into chromosomes. Each single chromosome is linked to its duplicate copy to make an X-shaped double chromosome. Now keep this in mind, once attached, each single chromosome is called a chromatid, one on each side of the X. Each double chromosome has 2 chromatids. Here in meiosis, prophase 1 includes 2 additional and very important steps: crossover and homologous recombination. Remember that the point here is to end up with 4 sex cells that each have just 1 single chromosome from each of the homologous pairs. But unlike in mitosis, where all the copies end up the same, here, every copy is going to be different from the rest. Each double chromosome lines up next to its homolog. So there's your mother's version lined up right next to your father's version of the same chromosome. Now, if you look, you'll see that these 2 double chromosomes, each with 2 chromatids, add up to 4 chromatids. Now watch. 1 chromatid from each X gets tangled up with the other X. That's crossover. And while they're all tangled up, they trade sections of DNA. That's the recombination. The sections that they're trading are from the same location on each chromosome. So one is giving up its genetic code for hair color or body odor, and in return, it's giving the other chromosome's genes for that trait. This is important, what just happened. Creating new gene combinations on a single chromosome. It's the whole point of reproducing this way. Life might be a lot less stressful if we could just clone ourselves, but then we'd also clone all our bad gene combinations, and we wouldn't be able to change and adapt to our environment. Remember that one of the pillars of natural selection is variation, and this is a major source of that variation. What's more, since all of the 4 chromatids have swapped some DNA segment at random, that means that all 4 chromatids are now different. Later on in the process, each chromatid will end up in a separate sex cell, and that's why all eggs produced by the same woman have a slightly different genetic code. Same for sperm and men. And that's why my brother John and I look different, even though we're both made from the same 2 sets of DNA, because of the luck of the genetic draw that happens in recombination. I got this mane of luscious hair, and John was stuck with his trash brown puff, and don't forget about my mad Assassin's Creed skills. Listen, metaphase 1, and in mitosis, you might recall that all of the chromosomes lined up in a single row, powered by motor proteins, and were then pulled in half, but not here. In meiosis, each chromosome lines up next to its homologous pair partner that it's already swapped a few genes with. Now the homologous pairs get pulled apart and migrate to either end of the cell. That's anaphase 1. Final phase of the first round, telophase 1, rolls out in pretty much the same way as mitosis. The nuclear membrane reforms and nucleoli form within them. The chromosomes fray out back into chromatin, increase force between the 2 new cells called cleavage, and then the 2 new nuclei move apart from each other. The cells separate in a process called cytokinesis, literally again cell movement. And that is the end of round 1. We now have 2 haploid cells, each with 23 double chromosomes that are new, unique combinations of the original chromosome pairs. And these new cells, the chromosomes are still duplicated, still connected at the centromeres. They still look like X's. But remember, the aim is to end up with 4 cells, so it's time for those sequels. Here, the process is exactly the same as mitosis, except that the aim here isn't to duplicate the double chromosomes, but instead to pull them apart into separate single-strand chromosomes. Because of this, there's no DNA replication involved in prophase 2. Instead, the DNA just clumps up again into chromosome, and the infrastructure for moving the microtubules are put back in place. In metaphase 2, the chromosomes are moved into alignment into the middle of the cell, and in anaphase 2, the chromatids are pulled apart into separate single chromosomes. Chromosomes, uncoiling chromatin, decreased form and cleavage, and the final separation of cytokinesis then mark the end of telophase 2. From one original cell with 46 chromosomes, we now have 4 new cells with 23 single chromosomes each. If these are sperm, all 4 of the resulting cells are the same size, but they each have slightly different genetic information, and half will be for making girls, and half will be for making boys.
Thanks for bearing with that. I know you might have to watch that again. That guy speaks really quickly, but it's good information. I'd just like you to read some pages in the bird book, the parrot book, and then make sure that you draw out these phases of meiosis, which you might have already. And then make sure to be able to explain these things. You're going to see them on the test, and they're really, really important because we're about to go into evolution, and this is a source of variation in populations on which natural selection works. Thanks for your attention, everybody.